Just a moment. I'm just fetching out of some questions to you.
Okay.
I'm not able to copy and print it out. Let me see how can I do it for you. Okay. Have you got the first question?
Yes. Shall I tell you the answers now?
Yes, please, you can.
Okay, so the first one, question 1AI, the term catalyst means to speed up a reaction by not being used up.
Okay.
Second part is activation energy is the amount of energy needed for a reaction to start, and in the presence of an enzyme, the activation energy is decreased. C, question 1, the lock-in key model is where a specific substrate is complementary to the shape of the active site of an enzyme. So only a specific substrate could fit to that specific enzyme.
Okay.
The induced fit model is considered to be better because substrates that are similar can fit into the active site and the enzyme will mold to for a better fit of the substrate. And last question, denature. Denature means when the enzyme has been affected where it can't break down or join substrates anymore.
Okay. Fine.
That's usually for pH, it's if it's become more acidic or alkaline, and for temperature, it's like above 37 degrees.
Mm-hmm. Okay, fine. So that was these questions. Now I think I have completed somewhat in enzymes. There is okay in enzymes. May I know what um, do you have a link with enzymes and food or enzymes in and digestion part, or it's only enzymes?
Sorry, could you repeat your question again?
No, I just ask like whether for your syllabus when we're talking about enzymes, is it only on the topic enzymes, or is it with enzymes and food, or enzymes and digestion like this? Anything regarding?
Um, Enzymes and digestion is
You
a
have
separate topic.
enzymes and digestion you all have, right?
Yeah.
Okay, so we'll do that. Fine, enzymes, okay. So we just spoke about enzymes, different things what enzymes does, and okay. So we'll just have a small recap of whatever enzyme actions have been done. Now we know that enzymes, they are proteins, right? They are proteins that act as a biological catalyst. When we are talking in terms of biology, they act as a biological catalyst. Fine. Then these enzymes, they work on the substrate. What do you understand by a substrate?
Yes.
What is a substrate?
The substrate is what fits into the active site. So what's going to be broken down?
Yes, a substance which acts on an enzyme, right? So these enzymes, they work on an substrate. Then the enzyme's active site and its substrate are complementary in shape, right? I think this also we saw last time. Active site and substrate, they are complementary in shape. Yes. Then an enzyme will work on one substrate, it's substrate specific. It is substrate specific. That means it will react with one specific substrate at a time. and enzymes and the substrate, they collide to form a enzyme substrate complex. Right? They form an
Right.
enzyme substrate complex. Then what happened once they form the enzyme substrate complex? Now we saw active site and substrate, they form a complementary shape. After that they become specific, enzyme substrate complex is created. Then the substrates break down, right? They break down and the products are released. Once after the products are released, what happens? The enzyme is now free so that they can add up to some other substrate, right? And this whole concept is known as the lock and key concept. Yes?
Yes.
Since we are going to talk about enzymes and digestion today, what we'll see is we'll see quite a few examples of the active site and the substrate and what product they will be forming. Okay, for example, if we have an active site like amylase, have you heard about amylase? This is,
Yes.
yes, what does amylase does?
It breaks down amino acid, not amino acid, starch.
Okay, it breaks down starch into
Um, into glucose and
simple sugars, glucose and fructose, right? So it is not only, it will break down starch, it will break, it is, sorry, is the complementary starch and will therefore only break down starch, right? It is going to break down only starch. Will protein and fat also include in this?
um, proteins are broken down by protease and
Protease, that means these are specific, right? So
yeah,
amylase is going to work only for starch.
starch.
Okay, so what we'll see is we'll see few enzymes, their substrate, and what products we will be. Have you finished writing down this part?
Just yes, I've written it down.
Down? Okay. So what I'll do is I'll make a column. Enzyme, substrate, and the products. If we have the enzyme, carbohydrates, my substrate is carbohydrate. Okay, and products formed here are simple sugars, glucose. Second enzyme is amylase, and we know it is breaking down starch. Breaking down starch into again simple sugars, also glucose. Protease, that is, it is breaking down protein into So here we have the enzymes part. These enzymes are breaking down these substrates into these products. Got it?
Yes.
Tell me once you're done.
I'm done.
Done? Okay. So we have something called as inhibitors. Now what are inhibitors? Now these inhibitors are molecules that partially fit into the active site. And because of this, what happens? Uh, the substrates are not broken down, okay? Because of these inhibitors, the substrates are not broken down. And only because they are not broken down, we do not have a proper rate of reaction. Just imagine if this is my enzyme, okay? Now, inhibitors will be something like this. Okay? It's just
Okay.
partially covering up the active site. So what will happen? Normally, I will be having another substrate which is actually waiting and coming to join here. Okay? But because of these inhibitors, they are unable to come and join over the active site. Because of this, the substrates are not broken down and the rate of reaction is also slowed down. Got it?
Yes, got it.
Then what else can we talk in enzymes?
Isn't there competitive and non-competitive inhibitors?
Competitive and
Non-competitive inhibitors.
okay, you want that also? Okay.
Yes.
Yeah. So we have two types of inhibitors, right? Sorry. One which is a competitive inhibitor and the second one is a non-competitive, right? First we'll see what is a competitive inhibitor. Now a competitive inhibitor is any compound which is closely resembling the substrate, okay, which is somewhat closely resembling the substrate. Now I'll show you with the help of a diagram. Now if suppose this is my enzyme over here, I have two active sites, okay? I have two active sites. Now I also have few substrates. Okay. Now these are the substrates which are found over here, okay? And this is my enzyme here. Competitive inhibitor means, if you see, these three have a close relationship with this active site, right? Some or the other is going to fit over here.
Right.
Yes, this is competition. It is like which of the substrate is going first and going to fit in it. Okay, this is a competitive
Okay.
inhibitor. Whereas if we see non-competitive inhibitor, it is completely opposite. That means I have an enzyme and I have substrates which are of different shape. So what is happening? Only a part of it is going and attaching to the active site. So it is a non-competitive inhibitor. But anything can go and get stuck to this enzyme. Okay. So this is the difference between a competitive and a non-competitive. Then we'll see the last part, I think, what we have in a digestive system. Are you done with this?
Just 1 second.
Yeah.
Yes, I did.
Now we were talking about digestive enzymes, right? Now,
Right.
enzymes are used in digestive system, as we know, to break the large, the complex, and the insoluble food molecules, right? Large, complex, insoluble food molecules are broken down into small, simple, and soluble molecules, right? Why are they broken down? Why are they not accepted as they are?
Because they can't be absorbed into the intestines.
More than intestine, they need to be get absorbed in the bloodstream. Okay, so that they can be absorbed in the blood stream, right? Suppose these are the complex carbohydrates which are present. Okay, they need to be passed on to the bloodstream. But how do they pass? Because if suppose they have a very thin permeable membrane like this, right? They need to pass through this permeable membrane. Permeable membrane won't have bigger space. It will have small breakdowns, permeable membrane, right? And how can these will pass through these ones? So it will be quite difficult, right?
Okay.
Use the enzymes so that they can pass through these walls. So these now large complex insoluble substances are, with the help of different types of enzymes, they are broken down into small parts so that they can easily flow from one end to the other end. Clear? Then we'll see what role has small intestine. Now small intestine, also called as the ileum. Now, food is digested in the mouth, right? Digestion starts from the mouth. From mouth, where it goes?
Sorry, could you repeat the question again?
Digestion process. Digestion starts with the mouth. From mouth, where does it goes? Next organ?
Um, well, it goes down the esophagus.
It goes down to the stomach. From the stomach, it goes down to the small intestine. Okay. Now, in the small intestine, with the help of three main enzymes, that is amylase, protease, and lipase, all my large complex insoluble food molecules are broken down, okay, with the help of amylase, protease, and lipase. So the main function of this small intestines or the ileum is the absorption of digested food particles. Digested food products into the bloodstream. Okay, why are they why is this happening? Because they need to be transported, right? You understood this large ones, they need to be broken down into the smaller ones so that they can pass through this cell wall. Clear? Now my next question is, why are they using only the small intestine? Why not these things breaking down in stomach, and why only in small intestine?
Um, uh, is it because the proteins and the simple sugar molecules are needed more in the blood to be transported to different sections of the body
No,
rather
nothing
than
like
being
that. Each and every organ you will be finding enzymes, the required enzymes.
oh,
Why
okay.
in small intestine? See, we know that digestion starts from mouth, right? We have saliva in
Right,
our mouth, which is breaking down, right? We chew our food. That saliva is getting mixed up. That also contains enzymes. So basically, from mouth, we have enzymes. But why specifically in small intestine does amylase, protease, lipase, or the absorption of digested food takes place? Why not in stomach?
um,
Okay, if you have a look at this, okay, this is mouth. If suppose this is a mouth, this is the stomach region, and from stomach region, how is your intestine? Is it small? Is it big? How can you say that?
um, it's got a large surface area.
Yes. Okay. Now, this is my stomach region. This is my intestine. Now, why specifically in stomach region? As you said, first point, it has a large surface area.
Oh,
Okay, because it's long and folded, it has a large surface area. So it takes time for these small enzymes to work up, right? Even if it is not happening
right,
in this intestine, it will slowly happen in this section, right? Therefore,
right.
large is one reason why this absorption is taking place. Second one is they are quite thin comparatively to the other organs. If we see stomach is one whole organ, it is quite heavy because and small intestine is long, so they must be thin, right? They are folded, and only because they are very thin and folded like this, right? So second point is because they are food does not have to travel to reach the blood. Okay. Third one, they are permeable. Now, permeable. What do you understand by the word permeable?
It's easier to pass through.
Easier to pass through. So what is happening here? Digested food can pass through them easily. Okay. Fourth point is they have a good blood supply. Intestine has a good blood supply. So to maintain the concentration gradient, okay, what do you understand by concentration gradient?
Moving from high concentration to a low
High
concentration.
concentration to a low concentration. So that is happening between the ileum and the blood stream. Okay. Fifth point is they have small projections called as villi, a finger-like projection. So if this is the intestine, they have small projections like this. Okay. Now what happens is these villi, they further increase the surface area. Now if this is the surface area and these are the villi, okay, what happens if I stretch the villi? If it is not like this, if I stretch, will it go more than my normal surface area?
They'll be larger.
It will be larger, and this also helps because already it has a large surface area and villi are adding an extra feature to it. Got it?
Yes,
Understood why specifically small intestine or ileum?
because of the large surface
Yes.
areas.
So these are the five main factors why they are involved. Done?
Yes, I've written it down. Thank you.
Okay. Now we have spoken about these five things, right? Which is helping us in small intestine. Now we'll see why specifically villi. What are the features of villi that is helping us in digestion or absorption? Okay. For that, I need to draw a diagram of villi. Okay, this is just one projection of the villi which I have drawn over here. Here is the lumen. This is known as the lacteal. Okay. Now, what are the features of villi? Now, villi has a large network of capillaries. You see the blue ones and the red ones? So they
Yes.
very good supply of capillaries. So once this blood becomes high in digested food products, what is happening is it is transported away and replaced with blood that is slow in digested food products. So because of this, there is a concentration gradient maintenance which is happening for diffusion. Okay, first point we
Okay.
see because it has a good blood supply. What is happening in this good blood supply? The villi has large network of capillaries. Okay, so this is the procedure of what is happening with the blood supply, how concentration gradient is getting maintained, and there is a diffusion between the ileum and the blood stream. Okay, second point we'll see is the role of lacteal. You see lacteal, this one, the light blue
Yes.
one. Now, this lacteal is a tube that is absorbing the products of fat digestion. So lacteal, does it?
Absorbs fat digestion.
Absorbs products of fat digestion. Okay, they absorb the fat digestion before turning them to blood. So before they are being transferred into blood, they absorb the fat. Okay, that is the role of a lacteal. Third, we have the single layer of epithelial cells, the orange one. Why is it necessary? This is reducing the diffusion distance. This is reducing the diffusion distance that is digested by the food products, and they have to travel in order to enter the bloodstream. So in order to enter the bloodstream, what is happening? They are just reducing the distance. So distance is reducing the diffusion distance. Okay, the last one is the permeable membrane. Permeable membrane. Simple one. Why? Because only if they are permeable enough, food can pass through them easily, right?
Right.
Okay.
Yes. I'm done.
Okay. Fine. Anything else needed to be done in enzymes?
I think that's all.
Any other topic which you have done in enzymes apart from these?
Um, that's all we've done up to at the moment.
Not
But
much.
no. Um,
So you can go through whatever we have done in enzymes today about enzymes and the digestive part, and ask me if you have any doubt regarding this.
okay. Do you have any exam questions on enzymes?
Yeah, I'm just going through that. I'll give you now.
Okay.
I've just sent you a link regarding a questions sheet.
Okay.
Okay. I think that you can do it for homework.
Okay.
Okay. So it has many questions regarding enzymes. Go through it. It is a bit of lengthy paper. Read the questions and answer them accordingly. Okay.
Okay.
So understood whatever we have done today for class?
Yes, I understood it today.
Okay. Shall we wind up over here?
Yes.
Yes.
Yes, thank you very much.
Thank you.